Hello guys, good morning everyone. Welcome to the class of biology on the fascinating world of biology. Hi, I am Nishad and I will be your instructor for this course. Through this course, we will learn about mitosis and their stages like prophase, metaphase, anaphase, telophase, cytokinesis, and we will also discuss here interphase and cell cycle. Here we will improve our reading skills. Now we discuss about mitosis. Mitosis in mitos. First of all, mito means equal distribution. Mito means equal distribution. Sis means process. In mitosis, a cell molecule divides into two identical daughter nuclei. Two identical means two same cell. Each daughter nucleus has the same number of chromosomes as the parent nucleus. For example, skin cells divide regularly to replace dead cells that have been shed from the surface of your skin. Your skin cells contain 46 chromosomes each. After mitosis, each daughter cell will also contain 46 chromosomes. Chromosomes are made up of the molecule deoxyribonucleic acid. Deoxyribonucleic acid. DNA stores all the information that a cell needs to grow and carry out vital activities. This information is stored as sections of DNA or genes. We say that the daughter cells produced by mitosis are genetically identical to the parent cell. Mitosis is a nuclear division such that the daughter nuclei produced contain the same number of chromosomes as the parent nuclei. Look at this diagram and try to understand the process of mitosis. Parent cells contain 4 chromosomes. It means a human has 46 chromosomes in cells. Here we look at 4 chromosomes 1, 2, 3, 4. And these thread-like structures are called chromosomes. DNA replicates means DNA duplicate. Mitosis process starts here, and we looked at two daughter nuclei. Two daughter cells are produced, which has the same number of chromosomes as the parent cells. 4 1 2 3 4 1 2 3 4 1 2 3 4. Two identical daughter cells are generated here. Mitosis results in two identical daughter cells. Look at this diagram and understand the process of mitosis. Parent cells DNA replicates and generate two identical daughter cells like parents, which has 4 chromosomes. Now we discuss about interphase. Cells spend very little of their lives in cell division. The resting or non-dividing stage of cell is called interphase. It means the pre-period of cell division is called interphase. Cells spend most of their time at interphase. Cells spend most of their times in resting form. Although interphase is termed the resting stage, interphase is also called resting stage. Cells do not actually rest. Cells still carry out activities such as absorbing nutrients and building up protoplasm. At interphase, chromosomes appear as long thin threads called chromatin. The chromatin threads cannot be distinguished individually at this stage. It is also during interphase that the centrioles divide in an animal cells. Centrioles are absent in plant cells. Look at this diagram. This is a cell, and these thread-like structures around the nucleus. Inside the nucleus, thread-like structures are called chromatin, and the outerboard boundary and nucleus between yellow substance are called cytoplasm, and the upper part tube-like structure is called centrioles, and these boundary line are called nuclear envelope because nucleus is inside this membrane. That is why called nuclear envelope. Animal cells at interphase. Interphase means resting stage. Cell in this stage, cell is prepared for cell division. Just before the cell divides, the chromatin threads DNA molecules replicates, producing two identical chromatin threads. The two chromatin threads coil and shorten to become chromosomes. Each chromosomes now consists of two identical DNA molecules called sister chromatids. The chromatids are joined at a point called the centromere. Here we discuss about the chromatin. Chromatin shorten, dense, collect its form chromosomes, and two identical parts of chromosomes is called sister chromatids. And now we look here about these diagram or graphs to understand the chromatin, sister chromatin, and centromere. That's a thread-like structure. It's a thread-like structures. Chromatin thread contains one DNA molecule. Chromatin here is present, and the center part dot-like structure junction is called centromere. Now then DNA replicate, DNA create identical thread, one parts and two parts in friends, one two parts and is called sister chromatids, and the center parts where these threads are joined is called centromere. Two identical chromatin threads called sister chromatid, two identical DNA molecules. How a chromosome is formed. Now we look here, chromatin threads coil and shorten. These threads coil and shorten dense to form a chromosome. Like the structures, here is a very thin structures and shorten, dense and coil to form a complete chromosomes, and the center part is called centromere chromatid, and these two parts are called sister chromosome, sister chromatids. Now let's go for details. DNA replications and mitosis must be precisely controlled. DNA replications and mitosis are precisely controlled in a dividing cell. DNA replication faithfully copies all the information stored within the chromosome. It means that all the information which are required are present in chromosomes. This ensures that daughter cells produced by mitosis contain all the sections of DNA genes needed for subsequent cell divisions and differentiations. The resulting daughter cells can be said to be genetically stable. You learned that a zygote cells divides to form an embryo. The cells formed as the zygote divides must be genetically stable for the embryo to develop normally. If an error occurs during DNA replication or mitosis, it will be transmitted to the daughter cells. This may lead to harmful changes to the genes and affect how the cells function. The embryo may not develop normally. In adult human, mistake made in DNA replication or mitosis may cause the uncontrolled division of cells cancer. In most cases, cancer can be fatal as the cell loses the ability to control it the way they divide as well as their normal function. Fatal means harmful, dangerous. For example, cancerous growths in the alveoli of the lungs will reduce the surface area available for healthy alveoli to carry out gaseous exchange. Growth is a permanent increase in size accompanied by cell division and differentiations to form tissues and organs. For growth to occur, cells divide through a process called mitosis. Interphase is the stage before a cell divides. At interphase, a cell carries out its normal activities of absorbing nutrients and building up protoplasm. DNA replications and centriole division take place during interphase. DNA replication is a process that has to occur without errors. If error occurs, it could lead to harmful changes to the genes and cause conditions such as cancer. A chair breaks easily if it has designed badly. This is the same for cell. If DNA is not replicated accurately, the mistakes could cause damage to cell. It means if things are created या make properly a suitable way then will be stable. If not appropriate ways to create and it's very dangerous and easy to destroy it. Interface process, the most time is taken in interface, and interface is a resting phase. In this stage, cell is prepared for division. Division mitosis takes at much time, and cytokinesis division or cytoplasm took a like this very small time. The cell cycle. Now we discuss here the cell cycle. The cell cycle. The order of events which occurs between one cell division and the next is called the cell cycle. The cell cycle consists of three stages. Cell cycle consists three stages. Number one, interphase or resting stage. Number two, mitosis, which is nuclear division. Number three, cell division, which is divisions of the cytoplasm or cytokinesis. Means cell cycle has three stages: interphase, resting stage, mitosis. In mitosis, nuclear division takes place, and cell division, cytoplasm and cytokinesis takes place. Now we discuss about the stages of mitosis. Stages of mitosis. For convenience, the process of mitosis can be divided into four stages: prophase, metaphase, anaphase, and telophase. However, it should be noted that such division is arbitrary and that the whole process of mitosis is continuous. The sequence that follows describes mitosis in an animal cells containing four chromosomes. Note that replication of DNA occurs during interphase. Cell at interphase. Microtubules are hollow protein structures found in the cytoplasm of cells. They provide structure supports and help to move substance within the cells. Now we discuss the stages of mitosis like prophase, metaphase, anaphase, and telophase. Here we discuss first stage of prophase. First stage of mitosis is called prophase. Chromatin threads condenses, coil, and shorten to become chromosome. Such chromosome consists of two sister chromatids attached at the centromere. The chromosomes are now visible under a microscope. Look like these structures. Prophase stage, cell 4 chromosomes. These are called centrioles, and these short structures are ester. Nuclear envelope. Centromere junction is called centromere, and two thread-like structure is called sister chromatids. One chromosome make a one chromosome. In an animal cell, structures called esters forms around the centrioles. Ester form around the centriole. That is a centriole, and structure ester is formed around the centriole. These structures are called ester. Esters are made up of microtubules. Esters are microtubules which are visible in electron microscope. The two pairs of centrioles move apart to opposite poles or ends. These two centrioles move in opposite direction. One move is air and other one is air. Here is other one. One centriole is here and the other one is here. The two pairs of centrioles move apart to opposite poles or end of the cells. The nucleus disappears. At this stage, the nucleus, these are the disappear means. In a late prophase, the nuclear envelope disappears. A spindle forms with the spindle fiber extended from one pole of the cell to the other. Now here spindle fibers are formed. These as centrioles, and this structure is called ester microtubules. These microtubes enlarge in size like this. Here it is called spindle fibers. Here it is called spindle fibers. These are these process taken place in prophase. This process taken place in prophase. Now we discuss the second stage of mitosis, which is called metaphase. Metaphase. Chromosomes line up around the equator of the spindles. The centromere of each chromosome is attached to the spindle fibers. Look at this diagram and understand the stage of metaphase. Chromosomes are arranged at the equator in vertical position. These spindle fibers are attached to the point of centromere. That point is called centromere. This first one is called in microtubule structure is called ester. Then when it is become a large thread-like structure, then it's called spindle fiber, making up the spindle. Now we will discuss about the third stage of mitosis, which is called anaphase. Each centromere splits. The spindle fibers pull the chromatids apart to opposite poles of the cell. Once the chromatids are separated, they are called daughter chromosome. Look at this stage and understand the anaphase. Spindle fibers attach to the centromere and pull them in opposite direction, half is sides and then the half in opposite sides. These are then called daughter chromosomes. Now we discuss about the fourth stage of mitosis, which is called telophase. Spindle fibers break down. A nuclear envelope forms around the chromosomes at each pole of the cell. A nucleus forms, the chromosomes uncoil and lengthen to become thin chromatid threads. In telophase cells, spindle fiber are breaks and two daughter nuclei are formed in the opposite direction. Nuclear envelope. This one cell is nucleus is also chromatid thread-like structure. Nucleus furrow. A furrow is found in two poles. Furrow. Now we discuss the stage of identical daughter cells. In telophase are found two daughter cells and their cytoplasm is also divided. Cytokinesis. This is the division of the cytoplasm. In animal cells, cleavage or furrows appear. These are cleavage or furrow. Appear in the cytoplasm between the two nuclei. Between the two nuclei. The furrow deepen and two identical daughter cells are finally produced. This furrow is formed and this furrow become deepen like division takes place here and the two result into daughter cells are formed which have their own nucleus and cytoplasm. Stages of mitosis. Look at this diagram and understand the process. Prophase, metaphase, telophase, one cell, Phoroform, two division takes place, anaphase and positive stage and formed a daughter cell. Now we look a overview of the topics which we have to discuss here. At this time, we discuss about stages of mitosis. We discuss about the stages of prophase, metaphase, prophase, metaphase, anaphase, telophase, cytokines. And we also understand the process of a prophase. We read about the prophase and look at the diagram to understand the process of prophase. Then we go to the process of metaphase. Metaphase, which is understand to the diagram of metaphase. Then we go to the stages of anaphase and look the diagram to understand the process of anaphase. Then we go to the fourth stage of mitosis, which is called telophase, and we look at the pictures and diagram to understand the process of telophase. Then we go to the cytokinesis, division of cytoplasm, and we look at the diagram and we understand that the how two data cells are produced here. Now we discuss about the mitosis in plant cells. Already firstly we discuss about the mitosis in animal cells. Now we discuss the mitosis in plant cells. Mitosis in plant cell is similar to that in animal cells, except that first one is centriole all absent in a plant cell. Centrioles are absent in plant cell. Cleavage of cytoplasm does not occur. In animal cells, cleavage cytoplasm takes place, and in plant cell, cleavage of cytoplasm does not occur during cytokinesis. Instead, a cell plate is formed between the two daughter nuclei, dividing the cell into two. The cell plate is formed by the fusion of small fluid-filled vesicles produced by the Golgi apparatus. Look at this diagram. Formation of cell plate during mitosis in a plant cell. In animal cells, furrow takes place and two daughter nuclei separate, and in plant cells a plate is formed between the two nuclei. A plate is formed, that is a plate formed between the nuclei and two daughter cells separate. Let's we discuss about the mitosis and stages of mitosis: prophase, metaphase, anaphase, telophase, cytokinesis. And then we discuss about the mitosis in plant cells, which are slightly different from animal cells. In animal cells, cleavage of cytoplasm takes place, but in plant cells, cytoplasm does not during cytokinesis. And we will also look at the animal cells have a furrow and denser, then become two daughter nuclei. And in plant cells, we here looked at the a furrow does not takes place, but instead a plate is formed. A cell plate is formed during the process during the mitosis, During the cell plate is formed during the mitosis and the two nucleus is produced which are separated by the formations of a cell plate and two daughter nuclei are formed. And I hope this lecture is very useful for those students which are very weakly in reading skills. In here our purpose is to motivate is to improve the reading skills of the learner and which is very useful for the learners. Now we will meet tomorrow in our next class in next topics so that we will wait here and watch my YouTube channel. Thank you so much for listening my lecture.